Okay, so let's continue. We'll talk about the concept of instantaneous Nyquist rate and the reflection artifact by interpolation. Okay, so this is part of the nonstationary time series analysis. So the main reference will be this paper back in 2016, and the material can be viewed as a continuation of what you learned from discrete time signal processing, the standard textbook by Oppenheim and Sheffield. So let's take a look at this non-uniform sampling and why is it important. So recall this is the ECG signal, electrocardiogram signal, and you can easily determine those peaks. Those are called R peaks. And you can use the peak-to-peak interval, which is called R interval, RI, to measure how fast your heart beats per cycle. And this is the unit of millisecond, which tells you that from the first cycle to the second cycle, it takes 707 T4 milliseconds. And if you just view this as the kind of the sample of the instantaneous period of how fast your heart beats, then that is the signal you get. That is at 447, you get one heartbeat. Another time, second time, you get the second heartbeat. Third time, you get the third heartbeat, etc. And you can view this guy, as the non-uniform sampling of the underlying heart rate that we have interest to recover. And how to get the heart rate or how to get the instantaneous heart rate? It is actually you get the R interval time series and you do interpolation. Very simple. And once you get the interpolated R interval, that is actually 1 over instantaneous heart rate. And IHR is instantaneous heart rate. And this is by the heart rate variability analysis protocol. Usually we use cubic spline interpolation. And then once you get cubic spline interpolation, you get a continuous function and you just resample it, for example, to 4 Hertz. And interesting information is actually roughly below 1 Hertz. And you know our heart rate is roughly 1.2 Hertz, or sometimes if your heart beat very fast, it's about 2 Hertz and so on. But anyways, the sampling rate is close to the spectral information we have interest. Another example is about respiration. Again, you have R peaks. And the height, as we discussed in the previous lecture, reflecting your respiration. That is, when you inhale, your chest is full of air so that you have a lower magnitude. But when you exhale, your chest has fewer air and hence the lower impedance, so that you have a higher magnitude. So if you, again, connect them, that is what we call ECG-derived respiration, EDR. And again, this is a non-uniform sampling because our peaks appear at non-uniform sampling times. And the interesting information is again below 1 Hertz and close to the sampling rate. The question is, then what does it mean by sampling rate? What does it mean by sampling rate? When we say sampling rate, usually we say sampling rate, okay, colloquially, and it is usually referred to some kind of how fast you sample a signal. But implicitly, when we talk about sampling, the sampling rate, the sampling is usually uniform. But right now, as I told you, this is not uniform. But I kept using terminology sampling rate. Then what does it really mean? Okay, so that will be something we want to define precisely in this short course, short lecture, and so on. But let's do some observation. Okay, so this is an airflow signal, and you can see this is the concept time frequency representation. You see, this is the fundamental frequency, fundamental component. That is the harmonic, and so on. Very good. So you get a lot of dynamics. And this is an EDR signal you get from the ECG signal. And you can see if you do a careful comparison, you can see, hey, you get a lot of very similar dynamics. And this is the time frequency representation of EDR. And what do you see in common in between these two time frequency representations? You see 0.4 Hertz, this guy appear, right? Very good. So this is fundamental component. But in the EDR, you see something slightly weird. there is another, seeming to be another component, right? But this component doesn't seem to fit anything of that of the airflow. And moreover, you see, I put a red dash in between. And what is this red dash? This red dash is actually instantaneous heart rate divided by 2. Just instantaneous heart rate divided by 2. And if you pay attention, something very funny appears. That is, you see, this curve, this extra curve, seems to be reflected of the fundamental component associated with the instantaneous heart rate. Seems to be a reflection, but why? Okay, so the answer that we will see in a bit is that it comes from the side lobe effect of the spline interpolation. Because we run spline interpolation. Because interpolation, something weird appears. Okay, now let's take a look at some theoretical analysis, and so that we know what's really going on there. So there's a very quick 1-minute review of Nyquist rate. What is Nyquist rate? Nyquist rate is actually the minimal sampling rate needed to accurately capture all the information in a signal without introducing aliasing. Okay, so this is kind of what you learn from signal processing. Suppose a signal is band limited and the spectrum is supported in between 0 and the big. Okay, and what is a Nyquist rate? Nyquist rate is here. It's B, and this is 2B. B and the 2B. Okay, and then what is Nyquist frequency? Which is Nyquist frequency is half of the sampling rate. That is, you sample the signal at this rate, and the half of it is called Nyquist frequency. Okay, so for example, if a signal contains frequencies up to 1 kHz, the Nyquist rate is 2 kHz. Okay, so yeah, Nyquist frequency is here, and sampling rate, Nyquist rate is 2 kHz. Okay, so well, this is a signal contains frequencies up to 1 kHz. This is B, and Nyquist rate is 2B, 2 kHz. Yeah, so this is B. Okay, this example should be put here, not here. This is Nyquist frequency. We're talking about Nyquist frequency here, but this is about Nyquist rate. Okay, so this example should be here, not here. Okay, but anyways, you can see there are 4 numbers, and they are related by half, double, and the things. And what is the key information here? The Shannon Nyquist sampling theory tells you that a band limited signal, that is a signal that has spectral limited has a spectral limited supported on 0 and the B. This band limit signal can be perfectly reconstructed from its samples if uniformly sampled, very important, uniformly sampled at or above the Nyquist rate. Okay, so you need to sample, in some sense, for each cycle, for the fastest component cycle, you have to at least sample 2 times per cycle. That is essentially it says. 2 points per cycle. That is what it tells you. Okay, and usually sampling rate, if it is higher than Nyquist rate, then we are good because you will have more than 2 points per cycle, so everything can be recovered. However, this framework is under uniform sampling, harmonic functions, and the traditional Fourier analysis framework. But we are talking about something like time varying frequency, time varying amplitude. What we have interest is related to how the signal oscillates from time to time. So we want to generalize it to our framework. What do we do? So let's do a very quick and simple observation. When we say sampling rate is c0 Hertz, what does it mean? We can actually view the sampling rate c0 as a linear function psi, c0t. And then what we do is we sample at tk such that psi of tk is k. That is, every time when this linear function hit integer, we collect 1 sampling point. And because this is a linear function, so you can easily determine that the sampling point will be tk, which will be k divided by c0. That is, if the sampling rate is C0, that means every second we will sample C0 points, which means 2 consecutive samples will last for 1/C0 second. That is assuming the unit is second. Okay, so that is what we have here. Okay, so based on this interpretation, we can actually generalize it to a non-uniform sampling. What we consider is, okay, fix one C and some epsilon, which epsilon is very small. That is, we want to somehow well control how fast or how non-uniform you can be. And we assume the sampling is not too crazy. Okay, then you take a C2 function psi, such that psi prime is greater than or equal to C and its derivative is less than epsilon psi prime. That is, it means this function is C2 and it doesn't change too fast. And it is actually kind of sufficiently large than C. Okay, then what is it? Then we call psi prime the instantaneous sampling rate. Instantaneous sampling rate. Okay, and the psi prime over 2 is the instantaneous Nyquist frequency, which is half of the sampling rate. Now, what we are doing here, because we want to define a way to quantify how often we sample the data, the signal. So the sampling points Tm will be satisfied by Tm is psi inverse m. That is, when your function psi, when the function psi, which is a monotonically increasing function, because C is positive, psi prime is positive greater than C, is a positive function, monotonically increasing. When it hits integers, then we say, okay, we will sample 1 point. So this is the notion very similar to instantaneous frequency. We just want to sample things, when we see, okay, when the system tells us, okay, this is a green light, you can do the sampling, But when do you encounter a green light? It actually depends on how psi evolves. Okay, so that is kind of a generalization from the traditional linear function to a general nonlinear function. And we can then define instantaneous sampling rate, instantaneous Nyquist rate, and so on. And what is the instantaneous Nyquist rate? So suppose now we have a function. This function satisfies the adaptive harmonic model. C1, C2 will be the lower bound and upper bound for simplicity. Okay, and it is also satisfies slowly varying condition epsilon. In this setup, the frequency or the spectral content is actually phi prime. And by similar notion as before, we say, hey, if now you have 2 phi prime, it's called the instantaneous Nyquist rate. So somehow what you have is phi prime, this is instantaneous frequency, double the frequency is instantaneous Nyquist rate, and that is your sampling rate, how often you sample things, and so on. So you can imagine, as before, if the instantaneous sampling rate is greater than instantaneous Nyquist rate, then technically we should be good. So it suggests that we need a sufficient sampling rate so that we can recover the desired information. And there is a whole bunch of theorem you can develop or extend starting from here. For example, how to fully recover the signal from uniform sampling points, right? You can use a sinc function, interpolation, and so on. But here, we try to talk about how it happens for cubic spline interpolation, and so on. So there will be some other topics that we can explore in the future. But here, we focus on cubic spline or spline interpolation. Okay, so to do so, just recall the fundamental cardinal spline function. What is it? It is actually something just, well, the nth order spline interpolation is a convolution of uniform samples with nth order fundamental cardinal spline function eta n, where eta n's Fourier transform satisfy this formula. And you can imagine, suppose it's hard to see it from here. Okay, but anyways, you can play with it and see that this is a definition of fundamental cardinal spline function. And now, the main theory that we can utilize to describe what's going on after interpolation and what we see in the time frequency representation is the following. Suppose the function again satisfies adaptive non-harmonic, so adaptive harmonic function, and now you sample the function f with instantaneous sampling rate psi prime, which is greater than 2 times phi prime. That is, the intuitive sampling frequency is fast enough. That is, you will have at least 2 samples per cycle, although the instantaneous frequency changes. And you denote the nth order spline interpolation as this guy. Then, what you can show is this f tilde n, this nth order spline interpolation result can be written as AT times all this guy. Okay, this looks kind of complicated, but actually is kind of the interesting part we want to say. For example, when k is 0, you see you get phi of t, this guy back, because cosine even function. But when k is 1, what do you get? When k is 1, actually what you get will be the reflection with related to the instantaneous Nyquist frequency. And you will have greater k and so on. And the ck will depend on n, and it decays actually quite relatively fast. And you will have some kind of order of epsilon approximation. That is why we need a slowly varying condition. And you can imagine the proof that is given here depends on Taylor expansion and so on. But, well, that tells you what we really get after interpolation. The interpolated signal itself already contains something strange. And the time frequency representation or time frequency analysis is just a tool that digs out all these artifacts, all this information. That is, we will view this guy as artifact. Okay? So let's see some simulation. And this is one example. You have time varying amplitude, and you have some kind of time varying frequency. And this is, if you have uniform sampling and the sampling rate is high enough, that is what you get. Very good. And on the left-hand side, you can see that comes from the sampling rate, instantaneous sampling rate here, and those are the sampling points. You can see those sampling points, they're kind of a bit non-uniform, but well, anyways. Then, if you apply synchronous squeezing transform, then what do you get? You get the fundamental component like before, but you can see one extra component and the several other components. Those components are the reflection. So actually what you get will be, hey, look, this signal seems to be very rich. It contains several different information, several different oscillatory components, but turns out that is artifact. It is not true. Those artifacts comes from interpolation. And you can run reassignment, which is another algorithm that we may discuss in the future. And this is kind of another variation of synchronous squeezing transform. And you see, you also catch all those artifacts. So this is a very interesting phenomenon. If you don't pay attention, you may get wrong impression about the structure or dynamics inside the signal. And this is another example, which tells you how about if we use different interpolation. For example, use cubic spline, you get this guy, this guy. If you use linear interpolation, you also get all this. So it doesn't matter. And you can try to use higher order spline, and you can get something slightly better. But when data is noisy, then you run into some other kinds of troubles, and we will not systematically explore it here. I just want to show you this kind of reflection, artifacts, and so on. And even if the signal itself is harmonic, as long as your sampling is non-uniform, and you count on spline interpolation to recover a signal, then you run into troubles. Okay, so you have two different interpolations and so on. And here is just because this is harmonics, right? Then you can expect that, hey, I can count on instant power spectrum to observe all the information. And yes, you are right. If you just do everything correctly, uniform sampling, then everything's good. But if you have cubic spline and so on, then you run into some troubles. And certainly there are some kind of another set of research that when the data is sampled non-uniformly, then we have non-uniform Fourier transform. And when you have non-uniform transform, if you directly apply non-uniform transform, these artifacts will be alleviated, but there will be another set of problems. And in particular, that works when the sampling rate is uniform. The instantaneous frequency is constant. When instantaneous frequency changes, then that needs to be modified, and there will be another topic to discuss in the future. So let's look at some real data. And this is the EDR signal, as I showed you, and this is the simultaneously recorded airflow signal, as I told you. And as you have seen before in another example, you get this reflection. Very interesting behavior. And this is an instantaneous heart rate. Okay, it is instantaneous heart rate. The signal is in the blue, and you can see there's a kind of one component and another component. And if you just plot instantaneous heart rate in the middle, you see, hey, look, there's a very beautiful, perfect reflection. And this reflection is the nature of interpolated signal. So normally you use multi-taper synchronous squeezing or multi-taper reassignment, you get similar result. You always see these artifacts. Okay, very good. So you see something nice. And that is a more example about EDR using reassignment, okay, or synchronous squeezing. I think this is a repetitive. But anyways, you see that's a kind of, oh, no, this is reassignment. Before I showed you synchronous squeezing. Okay, good. So, in summary, not only inference, synchronous squeezing, or reassignment, actually, all the other signal processing algorithms are impacted, even basic Fourier transform. Why? Because it's the nature of the interpolated signal. So you have to be very careful in the interpolation of the extracted features when you study dynamics properties, in particular when you touch this non-uniform sampling field, or if the sampling rate is close to the spectral content you have interest, then you may want to be careful, because the aliasing behavior in a non-uniform sampling field is kind of different of the aliasing you encounter in the Fourier domain. And the possible solution is you consider high-order spline, blending spline, which we'll discuss later, or maybe there are some other techniques. Okay, so we combine time frequency analysis with non-uniform sampling directly without using interpolation. That will be what I said. We can try to develop non-uniform time frequency representation and do everything directly. And that is actually something we will discuss in the future. That is something also very, very interesting. I think that